We finished glycolysis and gluconeogenesis, and now we are doing anaerobic metabolism, which is kind of similar, except for this is what the body goes through when there's no oxygen available. So there are two kind of pathways that we're going to go over. One is anaerobic fermentation, and this is what like yeast does. And then there's the Cori cycle and just standard anaerobic metabolism, which is what humans go through. So we're going to start with what yeast goes through. So starting with pyruvate. It's only 2 steps, one intermediate, and then it ends with ethanol. So first we're going to go from pyruvate to acetaldehyde, and the enzyme that is going to catalyze that is called pyruvate decarboxylase. And this might look a little familiar. If we zoom out, we can see that pyruvate carboxylase over here is what's used in gluconeogenesis. So instead of adding on that carbon, we're going to be taking one away. So acetaldehyde is our intermediate, and then the last step is just taking it straight to ethanol. And the enzyme that catalyzes this reaction is called alcohol dehydrogenase. And also in this step, we are going to be oxidizing NADH to NAD. This is important because it reproduces NAD+, which if you remember, we can zoom out to glycolysis, is very important because we need that as an oxidizing agent. So that is anaerobic fermentation, I will label it. And so this does not happen in humans. This happens in stuff like yeast. Lastly, we are going to go over what humans do, and it basically just takes pyruvate to lactate and back. And again, the goal is to reproduce that NAD+ so that we can keep glycolysis going. We're going to go pyruvate straight down to lactate. There's no intermediate in this step. This reaction is catalyzed by lactate dehydrogenase. And in this one, we are also going to produce an NAD+. So that's really the main goal. The pathway back from lactate back up to pyruvate uses the same enzyme, lactate dehydrogenase, but instead it takes 2 GDP and 2 ATP. Okay, so as you can see, we used 2 GDP and 2 ATP, and we turned them into GDP and ATP. Now I'm going to label this Cori cycle. And for the Cori cycle, from going lactate back up to pyruvate, we're not producing NAD+, so that's not the main purpose of this pathway. This is really just to prevent lactate buildup. So if you're exercising a lot and your body doesn't have enough oxygen, you're going to go through this anaerobic metabolism. And by doing that, lactate is going to build up in your muscles. This is often called lactic acidosis. So your pH levels drop, which is not really good. So bringing lactate back up to pyruvate kind of prevents that lactate buildup. The Cori cycle also kickstarts gluconeogenesis, which is going back up to pyruvate to glucose. And the purpose of that is just to make sure the body has enough glucose to go through glycolysis so that it can produce enough ATP, enough energy for the body for however much you're working out, expending energy. You need to be able to create enough energy, and to do that, you need a lot of glucose, which you get glucose by going through gluconeogenesis. So the Cori cycle kind of kicks off that pathway. That's pretty much all there is for anaerobic metabolism. The 2 main pathways for fermentation, which is like yeast, and that goes to ethanol, and then just regular anaerobic metabolism in the Cori cycle for humans. Just remember that a lot of lactate can lead to lactic acidosis, which lowers the blood pH, which is not good. And that can happen during intense exercise. That's pretty much it. All of the notes will be on the link tree, biochemistry, and then anaerobic fermentation. And then next time we're going to do the transition step.